Hey guys, just wanted to quickly go do kind of an overview on Chapter 4. I know that for the most part we have discussed cell structure and function on our Zoom meetings, but I just wanted to hit some of the highlights for you guys. So remember, and this is kind of one of those things that you don't want to forget, that cells are the basic unit of life. They are the basic unit of structure and function for everything that's living. So that means that everything that's living is made up of cells. Cells in general are pretty specialized. We have lots and lots of different types of cells, and their size matters. So the smaller the cell, the better they are at exchanging materials across that semi-permeable or selectively permeable plasma membrane that we've talked about. In general, the idea of studying cells came way back in history in like the 1800s when some scientists discovered that everything is made of cells and cells come only from pre-existing cells, and that they are the basic unit of structure and function for every living organism. Here are the scientists that are behind that theory, and it still holds true today. All of that research still maintained today. For us, where it's very critical that we understand that there are two types of cells. There are prokaryotic cells and eukaryotic cells. So prokaryotic cells do not have a membrane-bound nucleus and they do not have membrane-bound organelles, whereas eukaryotic cells do have a membrane-bound nucleus and they do have double membrane and, generally speaking, bound organelles. So prokaryotes are much more simplistic and smaller. When we study prokaryotes, we talk about bacteria and then archaeans as well. Eukaryotes do have that membrane-bound nucleus. They have membrane-bound organelles, and they include animals, plants, fungi, and protists. So the prokaryotes are, in general, much more simplistic, and we always use bacteria as a common prokaryote to talk about just because we know a whole lot about bacteria because we kind of needed to. So, and bacteria itself has many different forms. You're not responsible for memorizing those forms. They do have cytoplasm. They have a single coiled chromosome that is a DNA. It is just not surrounded by a plasma membrane, which is why it's a prokaryote and not a eukaryote. And prokaryotes are super small. They're way smaller than eukaryotic cells. So the domain eukarya do have that membrane-bound nuclei. Notice I've said this more than once because it's pretty important with regards to this chapter and with regards to your exam coming up. So this is an animal cell. Yes, you will memorize an animal cell. It's not too frightening, I promise. And a plant cell. So eukaryotic cells do have a plasma membrane around the cell, and it is a phospholipid bilayer. So we talked about phospholipid bilayers way back when we talked about chapter 3, and we were talking about the important biomolecules, and of course lipids were one of those, and so we pointed out why phospholipid bilayers were structured the way that they are, and really that allows for permeability, and it allows it to be selectively permeable. It makes sure that not everything can just go in and out of the cell willy-nilly. That would not be effective. So this shows you the plasma membrane and what it looks like. So you can see the phospholipid bilayer here. You can see that this is the hydrophilic or polar head. These are hydrophobic tails, and they orient themselves in such a way that the heads are pointing to either the cytoplasm or the extracellular fluid, which is real watery, and the tails orient themselves to each other because they are not fans of water. So plant cells do have a structure called a cell wall, and it's chitinous, so it's all about structure for a plant cell. Plants require a plant cell wall in addition to a plant cell membrane, because that cell wall keeps that plant from rupturing when that giant vacuole fills with water. Animal cells do not have cell walls. Animal cells do have a glycocalyx, and that's this sugary kind of complex carbohydrate thing surrounding the cells, and that is individual. It's a fingerprint for each of us. We all have our own individual glycocalyx, and that is one of the reasons why we cannot accept an organ from just anyone, because our sugars need to match or our body will not be friendly to it. So in general, the thought of why eukaryotes exist is that in fact, perhaps maybe there were simple prokaryotes and simple energy-related type of organelles that just got enveloped by that simple cell, and that created a much more complex cell. And it's really referred to as the endosymbiotic theory. We talk about this again a little bit later. So another very important piece to remember from this chapter is, of course, that there are two classes of organelles. So there's the endomembrane system and the energy-related organelles. So the endomembrane system consists of everything, all the organelles that actually communicate with one another, like the nuclear envelope, the rough and smooth endoplasmic reticulum, the Golgi apparatus, and the vesicles associated with those. And then the energy-related organelles are completely different. Those are the mitochondria and the chloroplasts. Those guys don't communicate with each other or other organelles so much like that. They're independent and self-sufficient. So the nucleus is the control center of the cell. It's one of the critical components of the cell because it does contain our chromosomes, which have all of our genetic information inside. In addition to that, there are also protein building components, which are, of course, RNA. Ribosomes are important for proteins. They're actually the site of protein synthesis, and you can find those in the cytoplasm or bedazzling the outside of the rough endoplasmic reticulum. The endoplasmic reticulum is basically the site of protein and lipid synthesis. So the rough endoplasmic reticulum is where proteins are folded and created, and the smooth endoplasmic reticulum is all about lipid production. Both the proteins and the lipids made in this rough and smooth endoplasmic reticulum do then get transferred to the Golgi apparatus for further manipulation and further modification. The Golgi apparatus is really the processing facility of the cell. So it has defined spaces, and it has basically the ability to receive and modify those lipids and proteins that were created in the ER. Essentially, once those lipids and proteins are modified, packages them up in vesicles. They're like kind of little pouches, and then moves those either somewhere within the cell or outside of the cell. Lysosomes are also produced by the Golgi apparatus, and they have like this little digestive enzyme situation going on, and basically they will envelop something inside the cell and then break it down if necessary, or if there's like an old organelle that's not functioning anymore, a lysosome will then break it down. Peroxisomes are super important. I remember talking about peroxisomes with you guys, and I mentioned that our bodies have the accidental ability to create hydrogen peroxide. However, luckily for us, we have peroxisomes that have an enzyme called catalase in them, and that catalase breaks down hydrogen peroxide into water and oxygen. Vacuoles are generally just very big membranous sacs. They're not found in all cells, but for plants, they're huge. They take up a huge amount of space in a plant cell. And so that large central vacuole conserves and holds water, and that's really useful for turgor pressure because plants don't have bones, and so they have to figure out a way to remain upright. And so what they do, they fill up their large central vacuole with a ton of water, and that presses against the cell wall of the plant cell, and then all the cells are doing that same thing in the plant, and they're all kind of pressing against each other, and it allows that plant to remain upright. If you come to my house, my plants do not have great turgor pressure because if they don't cry or bark or meow, they generally don't get watered. So they're all kind of sad and folded over. Mitochondria are referred to as the powerhouse of the cell, and the responsibility of the mitochondria is basically to convert sugars that we consume and make ATP, and mitochondria is very, very efficient at doing so. Both plants and animal cells have mitochondria, and essentially, we will talk a whole lot more about mitochondria and the job that mitochondria do once we get to a couple chapters ahead of us when we talk about cell respiration. Chloroplasts are where photosynthesis takes place in plant cells, only plant cells. There are some photosynthetic bacteria, like cyanobacteria, but it's more rare than it is common. So essentially, chloroplasts have these pigments called chlorophyll, and sunlight is essentially absorbed by chlorophyll, and then that sunlight that's absorbed by chlorophyll jazzes up a bunch of electrons, water is split, and then we have the ability to make some energy and basically use that energy to generate a sugar that's used for the plant. So chloroplasts have a three-membrane system. So the double membrane encloses the stroma that contains some important enzymes in there, and then the thylakoids form a third membrane, and those have chlorophyll in them, the important pigment. And those thylakoids actually are disc-shaped, and they get stacked on top of each other, and once they're stacked on top of each other, they're referred to as a granum. There are other pretty important plastids like chromoplasts that have pigments, and those pigments are responsible for the colors that we see in our leaves. Unfortunately, in the fall, chlorophyll is the wimpiest pigment, and so as soon as temperatures start changing and just start dropping, that chlorophyll will break down, and what that leaves you are those other pigments that are a little bit more rugged than the chlorophyll. So you'll see those carotenoids and flavonoids, and that's how you get this variation. Once chlorophyll breaks down and is no longer evident, then you'll see those yellows and oranges and reds and things like that. So another component that's pretty important for the cell is the cytoskeleton. So that is the internal framework of the cell, and it really helps to do a ton of different things inside of a cell. So there are three types of cytoskeletal elements that we discuss. There's actin, which is the smallest, intermediate filaments, which are the mid-size, and then there are microtubules. So actin is super thin, and actin is essentially used to move muscle. It has other jobs also, but the thing that we know most about actin, of course, that we think about more often, is that actin and myosin move and work kind of against each other, and they allow for motor movement. Intermediate filaments have various jobs, but they do a great job of supporting the nuclear envelope. They help form tight cell junctions where you don't want things to pass through those cells, and they give strength to skin. Those microtubules are the largest of the cytoskeletal elements, and they play a huge role in mitosis. They are responsible for moving chromosomes around during cell division. So, cell movement does also happen in some cells, essentially. Like, there's a 50% of the cells in the human body actually have a flagella, so they have a long whip-like singular string thing that essentially rotates like a propeller. And for humans, only there's sperm cells that have a flagella. Lots and lots of cells in our body have cilia, though. Those are shorter little hair-like extensions, and they generally line places that need things moved along. And so my example is always the fallopian tubes, where an oocyte has to be kind of swept along down the road, so fertilization could possibly happen. So we left off our actual Zoom discussion talking about viruses, and this comes from chapter 20, and it's not necessarily typical, but it's kind of something that's kind of critical for us right now, especially in the time that we're kind of dealing with. So viruses are associated with plant and animal diseases, but they are non-living. So it's kind of important that people have a better understanding that viruses are not living, but they are very, very small, 10 to 400 nanometers large, and they have tons of different shapes. Generally, they have two parts. They have a caspid, and then they have an inner core of nucleic acid. Here's some examples of various viral shapes. And what they do is essentially enter a host cell and inject their genome, and then the cells will begin to assemble new viruses, and they leave the cell through budding or bursting. And we tend to recognize that a virus has attacked, and so we employ all of our body's defenses. Our immune system will work really hard to fight off that virus. So, and that sometimes results in very small symptoms or even deadly symptoms like runny noses, fever, blood clots, even pneumonia. We can even attack our own infected cells to fight that virus that's infected us. Essentially, the flu and the COVID-19 virus has the tendency to cause immune system responses to attack the cells that line the lungs. If those cells that line the lungs are attacked, then they are not going to be allowing oxygen to diffuse like correctly, and so that puts you at an oxygen deficit. And if you're at an oxygen deficit, then your body's organs will be susceptible to sepsis. So essentially, the immune system tends to remember pieces of outer viral proteins. So those, if you are vaccinated, vaccines can inject pieces of the outer caspid so that your body recognizes it, but not necessarily inject the virus genome. Of course, some viruses mutate very quickly, and so that doesn't allow for permanent recognition of our bodies. Some people think that antibiotics should be able to work on viruses, but antibiotics work to break apart a cell wall of bacteria, and viruses don't necessarily have that cell wall and those kind of living organism components, and so antibiotics have no effect on viruses. The flu virus has a spike on it. They have a couple different spikes on its outer coat that the H spike allows the virus to bind to the receptors, and then the N spike attacks the host's plasma membranes, and so that allows mature viruses to exit the cell. And each type of spike can occur in different varieties. So our immune system only recognizes H spikes and N spikes that has been exposed to. New spikes um, are very concerning because they can potentially become pandemics, things that our body has never seen before. Emerging viruses are new or previously uncommon viruses, and there's a ton of examples of emerging viruses, and there's a lot of reasons why emergence of viruses can occur. So sometimes a virus may extend its range of recognition, like when West Nile was transported to the US and took hold in birds and mosquitoes. Or sometimes a genetic mutation can occur, like the various flu strains that were created through mutation of flu viruses, which had initially only affected animals and then transferred to humans. So it is necessary to get a flu vaccine every year due to the rapidly mutating flu virus. Coronaviruses are kind of unique, but we have all in our lifetime been exposed to various types of coronaviruses. So their spikes on the virus attach to different places in a body, but MERS, SARS, and SARS-CoV-2 attach in the lungs, and then the lung epithelial cells are destroyed, and sometimes immune systems become hyperactive in some individuals, and they create what is called a cytotoxic storm and destroy even more tissues than necessary. So what we're thinking about now, of course, is COVID-19 or SARS-CoV-2, because it's spread through droplets or aerosols, and that happens through coughing, talking, laughing. So it's tiny, 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 and it can land on surfaces. It can be inhaled. It can be absorbed through eyes even. So because COVID-19 is new to us and has a much higher infection rate than the flu, it's very concerning. And so even though lots and lots of coronaviruses are recognized by the body or not so detrimental to us because this is new, our body has no immune defense to it, and because it attaches in the lungs, that becomes problematic. And because of the way that it's spread, that's why we have to wear masks. That's why we're kind of protecting ourselves and each other, because our responses to this virus, some people are asymptomatic, they have no idea they even have COVID-19. Some people experience death from the complications of that cytokine storm or the cytotoxic storm, and their body just goes septic, and it's just catastrophic.